Welcome, and thank you for joining us today for our Virtual Investor KOL Connect segment featuring CNS Pharmaceuticals. My name is Janine Thomas. I am CEO of JTCIR, and I will be the moderator for this segment. Today, our focus is glioblastoma, or GBM, and with that, we will discuss the patient journey, the current standard of care, and the unmet clinical need. We will then focus on CNS Pharma's recently announced pipeline expansion with the in-license of TPI 287, a late-stage abiotaxane for the treatment of brain malignancies, and the expected development strategy. We will also touch on the company's ongoing, fully enrolled and designed to be pivotal study of its novel anthracycline berubicin in patients with relapsed and refractory glioblastoma. I am very pleased to be joined by Dr. Sandra Silberman. She is Chief Medical Officer of CNS Pharma. We are also joined by Dr. Don Picker. He is Chief Scientific Officer of CNS Pharma. And we have Dr. Samuel Goldlust. He is a leading neuro-oncologist and a key opinion leader who currently serves as a medical director of neuro-oncology at St. Luke's Health System and is a former investigator in the company's global potentially pivotal study of berubicin at his former clinical appointment. Before we get started, I want to remind our audience that CNS Pharma is publicly listed on NASDAQ and trades under the ticker CNSP. And during today's discussion, the company will be making forward-looking statements, and I encourage everyone to view the company's latest SEC filings on their website at cnspharma.com for the latest information. So let's get started. This is a roundtable format, so I encourage you to join in if you have anything to add to the discussion at any time. Dr. Goldlust, we'll start with you. You treat patients every day. Can you discuss the journey for GBM patients and the biggest challenges with the current treatment landscape?
Sure. The GBM is historically a very challenging tumor to treat. Unfortunately, despite the best efforts of neuro-oncology over recent decades, survival hovers around 18 months. Now, there's patients on either side of that median, but among the challenges that leads to that disappointing number is the lack of options we have for patients when their tumors recur. And survival at recurrence is somewhere between 6 and 9 months. And unfortunately, there's been no drugs to date that have shown to extend that interval. So I think a major breakthrough in the disease would really come if we had something to offer patients when their tumors recur. And the reason why some of the drugs we're going to discuss today are of particular interest, we know as a field that there are a number of drugs and drug classes that work in the petri dish to fight GBM cells, but the idea of getting those drugs into the brain at therapeutic concentrations has been historically very challenging. So if we have compounds that can cross the blood-brain barrier and get to the target where we need them to be, that would be a major breakthrough in survival. And of course, we're going to chat more about some strategies for that today.
Excellent, extremely helpful. So I'm going to turn to you, Dr. Picker. Can you provide an overview of TPI 287, how the molecule works, and how it gets across the blood-brain barrier and destroys tumor cells?
TPI 287 is an analog of a class of compounds called taxanes. Taxanes are one of the most widely used class of drugs to treat cancer in the world. An example is Taxol, which is the main drug to treat breast cancer and many other cancers. The one thing that taxanes can't do is to get into the brain. The reason that drug and many other drugs and other molecules can't get into the brain is because of a tissue called the blood-brain barrier that filters out any drugs or other foreign molecules transporting from the regular blood system into the brain. So these efflux pumps recognize all these molecules and pump them back into the normal blood system. It recognizes all the taxanes, so they can't get into the brain. What we've done with TPI 287 is to change the molecule so that these efflux pumps do not recognize the molecule any longer. It therefore cannot be pumped back into the regular blood system and can cross this blood-brain barrier and get into the brain. Once it's into the brain, it can then migrate through the tissue and get to the tumor. Once it gets to the tumor, it kills cells the way all the classical taxanes kill cells. And that is the molecule binds to the microtubule during cell division, stabilizes the microtubule so that it inhibits cell division. This kills the cell and so therefore operates like all the taxanes once it gets to the tumor cell. The big difference for this molecule is its ability to cross this blood-brain barrier and get into the brain.
Dr. Goldlust, you recently published an exciting manuscript about TPI 287 and its successful phase 1 study. Can you discuss the data from the study and what they mean for the drug's potential in GBM?
Sure. So it is a phase 1 study. So primarily we looked at safety first. We wanted to make sure that patients with recurrent GBM that the drug was well tolerated and safe as we went through the dose escalation. But our secondary endpoints were survival. And although the numbers are relatively small, we've only treated 25 patients to date. Compared to historical controls, we were very impressed with the preliminary data we've seen so far. And overall survival was around 13 months, and that's at the time of recurrence. As we talked about before, that number is historically somewhere between 6 and 9 months. So very promising in terms of the tolerability of the drug and gives us hope for future studies of the same compound that we can duplicate some of the numbers that we've seen early on. And we're working presently on developing those studies.
Great. So Dr. Silberman, I realize it may be early to discuss, but given the phase 1 data, can you describe your vision for the next phase of development for TPI 287?
Absolutely. We recognize that TPI 287 was similar to what our program in circles, which is those compounds that cross the blood-brain barrier and that can make a difference. So following on with berubicin, what we've done is created a network of investigators worldwide that have been working with us for recurrent glioblastoma. So we feel that the data from TPI 287 is exciting. It's already been discussed with the FDA, working on a pivotal trial to come next with a recommended phase 2 dose or phase 3 dose. And so we're ready to hit the ground running, and not only with the drug and the regulatory authorities, but we're established with our established investigator network.
So Sandra, I must say this is very similar to your development strategy with berubicin. It's taking a classic chemotherapy drug, modified to cross the blood-brain barrier, and testing it in a potentially pivotal study. So this does sound like your approach with berubicin. How does this dovetail with berubicin?
Absolutely dovetails quite well. We spent 4 years or more developing our investigator community, both in the United States and in Europe. And they were interested and excited about using this drug as an option for recurrent glioblastoma. Because of that investment in this community, we feel that TPI 287 can hit the ground running because we already have these sustained relationships and the interest in developing these kinds of compounds. So I feel that when we looked at TPI 287 and how it was developed, we realized this fit totally within the CNS pharma concept of drug development and our community. So I feel that this is going to be an excellent add-on for us, and it's an area where we have expertise.
So we've been hearing about an interesting approach to treating GBM using targeted ultrasound. Can you discuss your thoughts on this?
I can take that one, Sandra. So there is kind of a movement within neuro-oncology to design clinical trials using a few different technologies. One is focused ultrasound. There's another called convection-enhanced delivery, with the idea, as we kind of touched on earlier, that there's a number of drugs that we have seen work in the basic science lab to fight glioma cells and GBM cells but getting the drugs to where they need to be historically because of the blood-brain barrier has been a challenge. So these techniques, focused ultrasound, CED as we call it, they essentially mechanically bypass the blood-brain barrier, either by opening it transiently with focused ultrasound or using a catheter that actually penetrates the blood-brain barrier and goes directly into the tumor. You know, the interesting and potentially less cumbersome approach would be using drugs that are reformulated in the same class as taxanes or anthracyclines as we have with berubicin and TPI-287 to pass the blood-brain barrier on a chemical basis and get to where it needs to be without having to take those extra steps of the mechanical compromise of the barrier. So I think that the concept is actually relatively similar, is that we're trying to get drugs to where they need to be. In our case, in our purposes for these two drugs, you can give them intravenously and they can get to where they need to be, as opposed to sort of a more involved process with the same idea. So it will certainly be compelling to see when more data arises in terms of survival to serve as proof of concept for what we've seen early on.
So before we close, Dr. Goldlust, can you discuss your thoughts on the roles berubicin and TPI-287 can play in the armamentarium for GBM treatment?
Sure. So it's an interesting time for the field and for CNS pharma, in that we have potentially two drugs that might be efficacious for recurrent disease. And as these drugs continue in development, with the hope that they both show the signals that we're hopeful that they will in terms of survival, I can tell you that you won't have any complaints on behalf of neuro-oncologists if it turns out that we have two drugs that are beneficial in a space where, over the course of my career and all of my predecessors that trained me, we've had not much to offer these patients, aside from clinical trials in terms of something at the time of recurrence. So I have to leave it up to the folks at CNS to determine how the trajectory of the development of both of these drugs will proceed along. But I can tell you from the standpoint of a clinician, it's very exciting to see the data that we have to date, and it brings a lot of hope for me and to be able to share with my patients that these drugs are in development and getting closer and closer to the clinic.
And I'd like to add to that, CNS Pharma is developing the data set for this pivotal trial with berubicin. We should have that by early next year. And we're excited about sharing that data with the medical community because we feel that this will definitely provide them with an option, and then follow that on with the data from TPI-287. So I agree that having these two options for patients is going to be a major game changer for this field.
With that, this concludes our Virtual Investor KOL Connect segment featuring CNS Pharma. I'd like to extend a huge thank you to Dr. Silberman, Picker, and Goldlust for joining us today. And I'd also like to thank our viewers for your time and attention. As a reminder, CNS Pharma trades on NASDAQ under the ticker CNSP. And if you like what you saw today, I encourage you to visit cnspharma.com for more information on the company, to sign up to follow the company to receive their alerts, as well as follow their social channels to stay current on the latest information. You can also visit virtualinvestorco.com for a replay of today's event, as well as our latest segments and events calendar. Thanks, everyone, and have a great rest of your day.